So hi, before anything else, let me introduce to you myself. So I'm Alexander B. Albanil Jr. from the College of Development and Education of Central Bicol State University of Agriculture, taking up Bachelor of Secondary Education major in Science. So in this video, we are going to talk about the different stages of mitosis and meiosis. So without further ado, let's start. Mitosis is a process where a single cell divides into two identical daughter cells. During mitosis, a eukaryotic cell undergoes a carefully coordinated nuclear division that results in the formation of the two genetically identical daughter cells. Then, at a critical point during interphase called the S phase, the cell duplicates its chromosomes and ensures its systems are ready for cell division. The major purpose of mitosis is for growth and to replace worn-out cells. Mitosis consists of four basic phases or stages, which are prophase, metaphase, anaphase, and telophase. The first stage of mitosis is the prophase. So, during prophase, chromatin tightly coiled and form visible condensed chromosomes. Spindle fibers start to form. Centrosome move apart to the opposite poles of cell. Nucleolus disappear and nuclear envelope disintegrate. Chromatin in the nucleus begins to condense and becomes visible as chromosomes composed of two sisters chromatids that are pinched together at the site of the centromere. Special proteins attach to the centromere to form kinetochore. Finally, the nuclear membrane disintegrates and the nucleus disappears. Centrioles begins moving to opposite ends of the cell and fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle. So the next stage is metaphase. During metaphase, chromosomes aligns the metaphase plate. Spindle fiber fully form and centromere reach opposite poles of the cell. So during the metaphase stage, the spindle fibers align the chromosomes along the middle of cell nucleus by their centromere. This line is referred to as the metaphase plate. During this stage, the sister chromatids appear to repel each other, forming the familiar X shape of a chromosome. The organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. Next stage is anaphase. During anaphase, sister chromatids separate and move to the opposite sides of cell. Anaphase, enzymes break down the proteins in the centromeres, allowing the two chromatids to separate. Once the chromatids separate, they are considered to be individual chromosomes. The paired chromosomes separate at the kinetochores and move to opposite sides of the cell. Motion results from a combination of kinetochore movement along the spindle microtubules and the physical interaction of polar microtubes. So we are now at the telophase stage. So during telophase, chromosomes uncoil to chromatin. Nucleolus reappear. Spindle fiber disintegrate. Nuclear envelope reform. And the two identical daughter nuclei form. So during telophase, when the chromatids reach the opposite poles of cell, a new nucleus begins to reform around the chromosomes, forming two daughter nuclei. Inside the nuclei, the chromosomes begins to uncoil and the nucleoli reappear. Outside in the cytoplasm, the mitotic spindle disappears, having the centrosomes just outside the nuclear membranes. The chromosomes disperse and are no longer visible under the light microscope. The spindle fibers disperse, and cytokinesis or the partitioning of the cell may also begin during this stage. Once the nuclear membrane has reformed around the nucleus, mitosis is complete. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. Meiosis begins with a parent cell that is diploid, meaning it has 2 copies of each chromosomes or pair of homologous chromosomes. Meiosis 1. Meiosis 1 has 2 main purposes. Number 1. It is the reduction division, so it reduces the number of chromosomes in half, making the daughter cells haploid when the parent cell has diploid. 2. It is during meiosis 1 that most of the genetic recombination occurs. Prophase 1 stage. Just like in mitosis, during prophase, the DNA condensation occurs. The nuclear envelope and nucleoli disappear, and the spindle starts to form. The big difference is what is going on with the chromosomes themselves. As DNA condensation proceeds and the chromosomes first become visible, they are visible as tetrads, so tetrads become visible during prophase. Tetrad. Sister chromatids. Crossing over between non-sister chromatids. Chromatids after exchange. Meiosis 1 begins with a condensation of chromosomes in the liptotin during prophase 1. During the packaging, another stage of prophase 1, the pairs of homologous chromosomes align to form tetrads in a process called sypnosis. Corresponding segments of DNA of sister chromatids of 2 homologous chromosomes twist and cross, forming so-called chiasmata. In these regions, exchange of DNA between homologous chromosomes can occur. Crossing over. Crossing over or crossover requires that the chromosomes break and reconnect to the other chromosome. The next stage is metaphase 1. During metaphase 1, tetrads line up at the equator. The spindle has completely formed. It is during prophase 1 and metaphase that genetic recombination is occurring. Keep in mind that it only happens when there are tetrads. So as soon as anaphase 1 get going, genetic recombination is over. Next stage is anaphase 1. During anaphase 1, tetrads pull apart and chromosomes with 2 chromatids move towards the poles. So we are now in telophase 1 stage. During telophase 1, chromosomes with 2 chromatids decondense and a nuclear envelope reforms around them. Each nucleus is now haploid. Keep in mind that it is not the number of chromatids per chromosome that determine whether a cell is diploid or haploid, but it is the number of chromosomes and whether they are paired that determines this. Meiosis 2. So the purpose of meiosis 2, at the end of meiosis 1, each chromosome still had 2 chromatids, that is double the amount of DNA that a cell should have. So the entire reason to go through meiosis 2 is to reduce the amount of DNA back to normal. Basically, to split the chromosome so that each daughter cell has only one chromatid per chromosome, or the normal genetic content. As we go on through the phases of meiosis II, we will see that it looks just like mitosis. It is really similar to mitosis, so keep that in mind. The only difference is that the two chromatids per chromosomes are not necessarily identical due to genetic recombination occurring in meiosis I. So the first stage in meiosis II is the prophase II. During prophase II, chromosomes with two chromatids become visible as they condense, and the nuclear envelope and nucleoli disappear, and the spindle is formed. So the next stage is metaphase II. During metaphase II, chromosomes with two chromatids line up at the equator. The spindle is fully formed. All that genetic recombination primarily occurs during meiosis I. The way the chromosomes line up during metaphase II can also help to make unique daughter cells. Next stage is anaphase II. So during anaphase II, chromosomes split so that a chromosome with only one chromatid heads towards each pole. So we are now at telophase II stage. So in this stage, chromosomes with only one chromatid condense and get surrounded by new nuclear envelopes. The four daughter cells are now all haploid and have the right amount of DNA. They are ready to develop into sperm or eggs now. So that's all for this video. Thanks for watching. Bye!